Invitation and a kind introduction. As Alex mentioned, my research kind of sits at the nexus between ecosystem science and plant ecophysiology, although what I'm talking about today is definitely more the plant ecophysiology side. And I've worked in ecosystems that kind of span deserts and savannas and mangroves, sometimes on the scale of individual organs and sometimes on the scale of whole continental patterns. But the one sort of abiding theme, the common denominator that I always come back to is nutrients. And so today I'm going to take you to one of what I think are the most interesting and important frontiers for plant nutrient research, and that is the tropical forests. The work I'm talking about today is being done by a great group of graduate students and undergraduates in my lab. So I'm particularly going to highlight some work by a former master's student, Mia Marcellus, and PhD student, Caroline Dahlstream. And then with input from some great collaborators. So Ellie Good is a new hire in plant ecophysiology at St. Mary's University. You should definitely invite her for a seminar. She's wonderful. And also Monique Weemstra and Jennifer Powers in the US. So I'm going to start with the biggest picture. Nutrients fundamentally underlie productivity in almost every single terrestrial ecosystem. And that is true both in the present, but then they're also going to play a really pivotal role in the ability for systems to respond to rising CO2. So nutrient availability is a really big lever on the carbon cycle. Essentially, it boils down to all the carbon in the world is no use to plants if they can't access additional nutrients or perhaps increase their ability to use nutrients more efficiently in order to actually build more tissues. And our attempts to model nutrient limitation using things like this, these are Earth system models, these are the kind of models that the IPCC uses to predict climate, for example. Our attempts to build them are only as good as the understanding biologically that's going into them. And so good models need fundamental understanding of what controls plants' capacity to get nutrients before they can accurately estimate how much nutrients might be able to keep pace with carbon. And I will argue that nowhere is this question more relevant than in the tropics. So I don't need to preach too much to tell you that tropical forests, right, are powerhouses of net primary productivity. They represent a huge fraction of global plant biodiversity. And they're an interesting place to think about nutrient limitation because, and I'm speaking broadly here, temperate ecosystems, which tend to be limited by nitrogen, are quite different to tropical ecosystems that tend to be limited or at least co-limited in their productivity by phosphorus. And that's because the soils that we find in the tropics, they tend to be really old, they're super weathered, and a lot of their phosphorus has been locked up into biologically unavailable forms. And so what plants are left to survive on is a pretty small, readily available phosphorus pool, sort of in a simple chemical form, and a somewhat larger pool of phosphorus that's often bound up in kind of a variety of organic forms of increasing complexity. And those are going to become relevant a little bit later on. So the plant's search for nutrients in the soil has all of the makings of a good quest, right? You have an elusive quarry, a winding road, friends and foes, competitors along the way. And that medium soil is incredibly heterogeneous, right? So not just in the vertical sense, but on the horizontal scale. Soil is heterogeneous from scales of literally nanometers up to hundreds of kilometers, right? It can vary in its physical properties. So density, moisture, pH, the communities of microbes that live in it, the relative availability of different forms of nutrients. And as roots are trying to navigate this patchy medium, they're also frequently having to battle it out, right? So they're competing with other roots, with microbes for the same limiting resources. They're also having to fend off pests and pathogens, sometimes even cheetahs posing as symbionts. At the same time, they're having to build collaborations with beneficial fungi and microbes to gain nutrients. And this is all being done, right, by these organs that have to be extremely multifunctional. So the same structures that are seeking nutrients are also seeking water. They're also keeping the plant upright. They're also trying not to get eaten. They're also communicating with the biota around them. So as organs go, roots are incredibly complex and interesting. And so in order to manage these kind of myriad tasks, nature has evolved an incredible diversity of form and functions of roots, right? We think of plant diversity and we think of flowers and leaves, and I would argue that roots are just as cool. So plant roots express a really wide variety of trait values, and that can be both sort of across different species, but then even within a given species. And these traits span allocation, right? Roots are indeterminate organs. Plants can choose to grow more roots, allocate more biomass there. We see variation in morphology, so sort of a spectrum from like a thin, high surface area root through to something thick and woody that can have a huge variety of different architectures. And even sort of really fun, specialized structures, these, they're relatively rare, but these like fuzzy little bottle brush roots that are incredibly metabolically active occur in some species. They're also actively manipulating the rhizosphere around them. So roots can exude things like phosphatase enzymes, for example. So phosphatase enzymes break down some of those complex organic P forms we talked about to make them available for plant uptake. They exude organic acids, they can control the redox environment, the water environment of the soil around them. Symbionts are particularly important. So roots can form relationships with 2 to 3 major groups of fungi that can do things like increase the surface area for foraging or actively break down nutrients from more complex forms. Those are mycorrhizal symbioses. And then there's also 2 major associations that plants form with nitrogen-fixing bacteria. So that take nitrogen that's normally not available in the air and turn that into a biologically available form. On top of that, they often have a really distinct microbiome around the root. They can express different like transport systems on the molecular level. And then all of these traits, right, they vary in time. There's sort of rates of root turnover, and then they also have phenological patterns, sort of seasonally, the same way that you would see in leaves, for example. Okay, so I've laid out this kind of range of challenges and this smorgasbord of traits that plants potentially have at their disposal. But as with everything in biology, there is no perfect root for every environment. There is no root that can do all of these things, that can optimize all of these traits at the maximum rate at the same time. And this leads us to the question of like, what shapes which combinations and values of traits that plants employ? What controls the particular combination of traits in a given root, in an individual plant, in a species across a given environment, maybe over time? And to get at the contributing factors there, we think about the role of evolutionary history, of trade-offs, so kind of taking an economic perspective on investment, and then in plasticity, in shaping that trait expression. And you can imagine that the answers to that question, right, they address basic ecology. So things like competition and coexistence under limiting resources. From an evolutionary perspective, they sort of address the fit and the selection between an organism and its environment. Practically, we're often interested in the responses of plants and plant traits to global change in the future. And then also in an applied context, these types of traits are relevant for things like selecting species for tropical reforestation programs, for example. The root caveat that we always have to put in these kind of talks is that assessing what roots do is not as straightforward sometimes as you might hope. Unfortunately, they pretty much never grow above ground like in this picture. Instead, right, measuring them is often methodologically challenging. It often requires destructive sampling. I haven't personally had to resort to bulldozer science just yet, but I have not ruled it out in the future. I have colleagues who have done this. And that means that the data coverage that we have, sort of taxonomically, geographically, bias against the tropics, really vastly trails the data that we have for other plant organs. And we also have a real mismatch of like much more heavy trait coverage for certain easier to measure traits than for others. And in most organisms, the particular trait combinations or the values that we see, right, are shaped by their evolutionary history. And in fact, we see that phylogeny does explain a significant amount of the variation in the relatively limited number of root traits for which we have widespread data. So these tend to be kind of basic morphological traits, and then also root nitrogen, which is sort of generally considered to be like a proxy for physiological activity. And that phylogenetic imprint is true at large taxonomic scales. So like all seed plants, family and above kind of level. But a few years ago, this really important study came out. Hafiz in your department wrote a really nice commentary on it that found that about a third to half of the variation that we see in these three structural and nitrogen trait is explained by sort of family and above phylogeny. And very little of that variation is explained by climate, which is kind of surprising. We probably would have expected climate to be a bigger driver. Whereas if you contrast that with leaf traits, there's also a phylogenetic signal, but climate is much more important. Other traits we also see cluster out. So the types of these major different mycorrhizal associations tend to cluster phylogenetically. Nitrogen fixation is another example. Most fixing plants occur within one plant family, the Fabaceae. And there's also a role for resource strategy trade-offs to determine trait combinations. So you can imagine in the simplest hypothetical case, a given species could have any combination of trait values for any given trait, or essentially that the potential multidimensional trait space is extremely large. But we know, right, that doesn't happen in practice. And plant biologists have been looking for a really long time at how trait trade-offs play out, particularly for leaves. And in leaves, we've identified this kind of largely unidimensional spectrum where we go from long-lived, low-nitrogen, structurally expensive leaves to like a fast-lived, high-nutrient, fast-slow trade-off axis. And we do see some of those similar patterns emerge below ground, right? So in both leaves and in roots, there is a trade-off between like nitrogen content and lifespan. But roots get a little bit more complex. Based on some of those most commonly measured traits, in roots, we see the emergence of at least two major axes of variation. So you have a sort of a fast-slow axis, but then on top of that, there is also a DIY versus outsourcing axis, where plants can either grow more of their own roots, or they can invest more in symbiotic fungi to explore the soil for them. And then there's probably even additional variability that can be mapped on top of that. Along with trade-offs, there's also potential here for some complementary functions. And a special case that is currently debated in the literature, which makes things exciting, is for nitrogen fixes. Nitrogen fixes, and in the tropics, we're like predominantly talking about legume trees. So these are things in the family Fabaceae, you would recognize as, you know, the iconic African savanna tree, or like a lot of rainforest trees. Nitrogen fixes shouldn't be limited by nitrogen, right? They should be able to satisfy their nitrogen demand. And if they weren't already, they would then become potentially limited by phosphorus. But having that extra nitrogen available should allow them to be able to afford to build more phosphatase enzymes, which are nitrogen expensive to build, that can then access more organically bound phosphorus in soil. And then finally, root traits, or at least again, once the limited range that we've looked at, are plastic in response to all sorts of environmental factors and treatments, so things like nutrient levels, drought. And it's reasonable to assume, although it hasn't really been rigorously tested, I would say, that it's likely that some traits can be more plastic in response to environmental drivers than others. So when I started my lab at McGill in 2020, right on the eve of the pandemic, we have been focusing on looking explicitly at the role of each of these drivers. So phylogeny, coordinations and trait trade-offs, and plasticity in shaping the root traits that we see in tropical trees. And today I am briefly going to go over some plants that some projects that we have running in the lab that tackle those questions. So I named up the Fabaceae a couple of times already. You might also note that my lab logo is an acacia. And the Fabaceae is a really big plant family. They exist all over the world, multiple biomes, and they contain almost all of the nitrogen-fixing tropical trees. The ones that don't, they have a different type of symbiosis. It's a bit more rare. But not within the Fabaceae, not all plants can fix nitrogen. Only some of them can. There's a mixture. And Fabaceae are very important components of tropical forests. They're often very abundant. They're functionally important. So that nitrogen fixation plays an important role in like secondary succession, for example. And when we model things like climate with Earth system models, nitrogen fixes are treated as a distinct plant functional type in those models. And then finally, as I mentioned, they're proposed to have some interesting interactions between nitrogen and phosphorus. And all of those things make them an interesting system to ask about trait organization. And so people have questioned whether there are distinct functional properties related to this ability to fix nitrogen, right? You produce your own fertilizer, that's kind of crazy. And we have run into some contention there because some people have found that the fixes and non-fixes are different, some haven't. But virtually all the studies that we have are comparing fixing Fabaceae trees, not all of them fix, with other unrelated species in other families. And this has bothered me for a while because it seems like it's a real conflation of phylogeny and function. And maybe the conclusions that you come to kind of just depend which species you're actually comparing when you're trying to see how different they are. So are the signals that you see because of fixation, or are they because of other conserved traits that just happen to occur in the Fabaceae family? And I started down this road a little ways during my postdoc, where I was looking at a very small number of species. This actually sort of emerged by accident from another experiment. So a note for graduate students there is that sometimes you can very convincingly sell an idea that was not the idea that you set out to test initially. Where I had both fixing and non-fixing, well, one non-fixing Fabaceae, and kind of saw hints in my data that there was variation in the nutritional traits that were more related to how closely related species were than to their nitrogen fixation ability. So I had sort of two closely related fixes, and then these two were more closely related to each other, but one fixed and one didn't. And the traits sort of seemed to cluster out more by phylogeny than by functional type, although I was not obviously able to test it with this setup. And that was interesting as well, because that big study that Valverde-Barrantes proposed, in that he proposed that phylogenetic controls on root traits were likely to be significantly weaker at smaller taxonomic scales than at these like big kind of multi-family clade comparisons. And they actually haven't really been looked at very much at these particular like lower taxonomic scales. So the first experiment that we did in the Soper lab was attempting to control as much as possible for that really big conflation of phylogeny and function by looking within the Fabaceae at root trait expression in fixing and non-fixing relatives. To ask explicitly, is there a phylogenetic signal in root traits? Not at those core scales, but when we look within closely related species, do they differ by function, whether or not they fix nitrogen? And then this is also an opportunity, right, to explore more some of those coordination and trade-offs between traits. And we got around the issue of potential plasticity by growing all of these species in the greenhouse in like a common garden setup. So we eventually managed to source and grow in Montreal in the winter in a pandemic 22 species of tropical Fabaceae tree seedlings. These were functionally diverse, so they spanned multiple continents, Africa, Australia, but they were all within the same subfamily of Fabaceae. One thing that you might notice is that even with this design, we were not able to fully remove the conflation between phylogeny and function. There are a small number of kind of non-fixes that are spread through this part of the tree, but it was difficult to track down seeds to like really do an ideal kind of experimental design to separate those things out. But nonetheless, these are fairly closely related species that fall into those two groups. And there were quite a number of them. And then within these, right, we have multiple species within the same genera and then species in more distantly related genera. And we measured a variety of physiological, morphological, symbiotic traits to try and capture the full spectrum of root function, as well as some above ground traits that capture kind of growth and carbon supply. And also I'd say what makes this study kind of a bit more unusual is that it's fairly rare for studies to measure all these traits under the same conditions at the same time due to kind of logistical considerations. So it's quite the effort. And the first thing that I was maybe a little bit surprised by is that there is really wide variation in trait expression, even within closely related species. So if you're not sort of used to looking at these scales, I can tell you that there's quite a lot of variation. Here I'm showing you, this is the length of root constructed per unit mass, so sort of how long and skinny and thin roots are. Root respiration rates, the activity of those phosphatase enzymes, and they're quite variable. You will, or I can tell you as well that all of these traits, almost all the root traits showed quite a strong phylogenetic signal, right? When we're looking like Blomberg's K, Pagel's lambda. We also see, you can kind of, if you squint here, you can see there's a bit of mirrored patterns across different traits. So we did see coordination between traits that I'll get to in a minute. And then also, maybe most obvious here, that if you sort of squint, you can see maybe there's a bit of a difference in trait magnitude between fixes and non-fixes. And so if you plot all of these traits as principal components, we do see a separation in trait space between fixing and non-fixing Fabaceae. This linear discriminant analysis, functionally it's saying like if you, based on the traits of a given species, how effectively could you guess whether that species was a fixer or not? And the answer is pretty well, so there's good separation. And if we compare the trait means across groups, so here I'm plotting trait means. When we analyze these, they were phylogenetically corrected, right? So there's obviously phylogenetic conflation in just measuring means without considering that some species are more closely related. And in fact, the significance changes depending on whether or not you consider phylogeny in those analyses, which I think is a strong argument that, right, we should be incorporating this into plant comparisons more maybe than we are. But the above ground traits were fairly similar, right? There's no like clear emergent patterns of differences in above ground traits between fixes and non-fixes. What we did see was larger differences emerging below ground. So for example, the roots of fixing species tended to have a higher specific root length. They tended to respire more. They tended to have a greater phosphatase activity. And so bearing in mind that there's still some conflation, once you've done these phylogenetic corrections, it's interesting to see that there are still significant patterns. If we return again to that kind of multi-trait space, we saw the emergence of one axis here that's primarily related to above ground and leaf traits. So photosynthesis rate, growth rate, with some kind of foliar traits. And then a second axis that was describing root physiological traits, so an independent axis, where we saw that roots that had a high nitrogen content tended to respire more, have a higher enzyme activity, be thinner. And then finally, a sort of third independent sitting out here by itself for arbuscular mycorrhizal fungi, or the degree to which those plants were creating symbiosis with these fungi. And then finally, this is the most exciting one in my opinion, to address that big nutrient trait coordination question in fixes, people had previously mostly compared phosphatase activity between non-fixes and fixes as groups. and varied in their conclusions, I think, depending on who was in the groups. But here we found a continuous relationship, right? Not just between the presence or absence of fixation, but actually plants that fixed more nitrogen, or species that fixed more nitrogen, also had greater root phosphatase activity. And this sort of hints maybe at like a mechanistic relationship where plants that have more nitrogen, need more phosphorus, are able to invest more in creating those phosphatase enzymes. And even actually after you do like a regression that corrects for phylogeny, that relationship gets even a little bit stronger. So to conclude what we found there, within the Fabaceae, within one subfamily at that sort of relatively small taxonomic scale, trait expression is shaped probably both by phylogeny and by fixation ability, even if those things are a little tied together. And there is evidence for trait coordination, right, with functional implications for how Fabaceae might be able to access nutrients, particularly in these phosphorus-limited tropical ecosystems. So I'm going to switch gears a little bit to some theory. This was also pandemic science. A shout out to my PhD student, Caroline Dahlstream, who did the whole first chapter of her PhD remotely from Patagonia. And okay, so we talked about those two proposed axes that kind of describe variation in a handful of root traits. And we were interested in whether there was evidence in the literature for kind of coordination of a wider variety of traits. Like, do you tend to see the whole spectrum of root traits organized into particular syndromes? And because we know, right, that trait expression is likely to change to some extent with environment, we limited our scope specifically just to very low phosphorus tropical forests when we were looking for this evidence. And so we started with those two axes as a guide, essentially thick, thin roots, mycorrhizal to non-mycorrhizal. And then she also added in a dimension of categories based on the major mycorrhizal type. So whether species have ecto or endomycorrhizae, they vary in kind of their metabolic capacities. And so she dove into the literature, looked at a whole suite of traits, and there were not many cases where the same traits had been measured at once, right? So she could sort of fill in a few, and then she looked for areas where there were other correlations, other studies, and tried to fill in what she would predict about a whole suite of other traits based on these associations. And for a given syndrome that I'll explore in a second, to infer whether we think those trait values are likely to be low or high. So I'm not asking you to look in detail at this table, but when you summarize this table, the kind of sum arrangement of all of those traits across 5 strategies it shook out as, we see that she then tried to tie that to the particular soil phosphorus forms that those traits should theoretically be able to access. So as phosphorus increases in complexity, it becomes more difficult and more costly in carbon terms for those forms to be acquired. And what Caroline found was that these different strategies, you would expect them to maybe be more well suited to accessing particular forms of phosphorus of different chemical complexity. So for example, a thick-rooted arbuscular mycorrhizal species, predominantly she'd expect would be good at taking up phosphate, and a thin-rooted ectomycorrhizal species would be better at taking up sort of simple organic phosphorus. A non-mycorrhizal species might rely a lot on root exudates. In addition, she found evidence that species with all of these strategies seem to co-occur, or they could all be found in tropical phosphorus-poor tropical forests. And because they co-occur, and they might be suited to uptake a variety of different phosphorus forms, she proposed this as a mechanism for niche partitioning of soil P. So you can see here, these are our 5 strategies. Here is sort of soil phosphorus of increasing complexity, and we might expect different places on the spectrum to kind of be better at taking up different forms of P, which, right, if we go back to like a classic problem in biology that people have been thinking about forever, why do species co-occur? And particularly, how are the tropics able to support such high levels of plant diversity? So here's a neat mechanism. Unfortunately, testing this in the field It's likely to be extremely difficult. Certainly we have not come up with a good idea for how you might test this in the field. But instead, we're going to start in the greenhouse. So we have an experiment that we're setting up right now to test this, where we are planning to grow species with contrasting syndromes, so that sort of fall into those different categories, and to grow them either alone or in competition on a variety of soil phosphorus forms of kind of increasing complexity, to test the hypothesis that fine root trait syndrome diversity would permit soil phosphorus partitioning and would facilitate species coexistence, or essentially kind of predicting that that some species will grow better than others on different forms, and that they will grow better in combination on a mixed source where they're able to partition niches. In addition, this is a nice opportunity to be able to evaluate trait plasticity to different phosphorus forms, looking at maybe a wider variety of root traits than we would usually be readily able to measure in the field. And speaking of plasticity, right, in biology, we often, not arguing always, but I would say it's fairly common for us to think of the unit of variation as being an individual. And so when we think of variation in populations of plants, individuals and populations across a gradient of climate, maybe a gradient of fertility, at scales often, right, of like hundreds of kilometers. And that has been true of a lot of the way that we've looked for variability in root traits. But we can ask ourselves, like, where actually is the variation in these environmental factors that might drive the variation in plant traits? Right, at big scales, climate, for example, shows big imprints on like leaf traits. But soil can be heterogeneous at smaller scales. So we know that fertility varies at big scales, right? You know, from temperate to tropical biomes. Here's a map of soil nitrogen for all of Panama. But some of that heterogeneity can occur at really small spatial scales. And if we assume that roots should be highly plastic organs, how do the scales of variation in the environment and the scales of variation in the root traits match each other? Plants, of course, are also modular organisms, right? So each branch of a root is developing somewhat independently. We know plants have evolved, right, to exploit patchy resources. So the question is, how much does the development of each root on a plant actually vary independently? And surprisingly, we don't actually have a lot of data for this in natural systems, particularly for trees, right, which can have large root systems. You know, their root systems can span many meters. In temperate systems, this study, also with a collaborator of ours, came out a couple of years ago. They looked at Douglas fir in Canada across a climate gradient. And then within that, they looked at specific sites, blocks of soil, and individual fine root branches. They didn't trace the individual fine root branches back to individual trees, unfortunately, because that's quite difficult to do. But they found that most of the trait variation in 4 out of the 5 traits they measured was greatest. Like, most of that variability was happening at the scale of individual fine root branches, which I think was surprising at the time. And we took this question then to the tropics, and oh, we did trace fine roots back to individual trees, for better or worse. And we were trying to explicitly examine the spatial scales of variation. We might expect, right, the traits, the soil variables to vary at different scales than temperate systems. There might be different soil variables that are important, which is why it was kind of interesting to ask that in a tropical context. And we had this nested sampling scheme where we had 3 sites along a phosphorus gradient in Guanacaste in Costa Rica. This is also where Alex works. And then within that, we had a number of trees at each site. And then within that, we traced 3 different individual root branches from a tree, and we paired that with soil samples. And also with rhizosphere community samples, although unfortunately, some of the extractions failed, which we're very sad about. But this is brand new data. I was just texting with Caroline about this last night. And And so far, looks very interesting. So what I'm showing you here, she did variance partitioning analysis to say how much variance was explained at like regional, individual, or root branch scales. And for the soil characteristics, some of them varied at big scales, right? The available phosphorus in the soil, things like bulk density, cation exchange. Available nitrogen tended to vary more at the level of individual trees. But then for their root traits, root phosphatase activity did mirror like big regional gradients of phosphorus. But some of the other traits that she's measured, things like diameter and root length, so root morphology, root respiration, we have data coming soon for mycorrhizal colonization, tissue chemistry, those things, by far the greatest amount of variation was happening within individual roots of a given tree. And so that tells us something biologically, right? That there is plasticity within the modular parts of these organisms that can be really large for certain root traits. But in terms of more broadly how we study traits, like, so what? This is not really so much of an issue for us when we're looking in the greenhouse where our soils are like quite consistent. But root tracing in the field is difficult. And often what we're doing to understand how roots are behaving at a location is going in and taking a number of mixed species, mixed individual root cores. We're often taking a mean of a particular trait. And then looking, for example, across a big environmental gradient, say in climate, and concluding that oftentimes those traits are not responding to that given variable. And that shapes our thinking maybe about the capacity, say, for a root trait to respond to global change. But if in fact what's happening is that the variation is responding at really small local scales, those traits can be plastic. It's just that we washed out all the variation in the way that we've sampled them. So this certainly is not going to make our lives easier, but it's important to think about how we interpret the data that we find. So to bring this all together, I think from a biological and ecological perspective, this type of research can help us better understand tropical tree functioning in terms of, say, balancing the acquisition of different nutrients, maybe thinking about mechanisms of coexistence in tropical forests. But as I hinted at the beginning, another dimension of why these questions are interesting is that nutrients are a big lever on global climate models. And those models, as they're being built, right, they have to make decisions about how they're going to represent plant nutrient acquisition. Interestingly, we're at a phase right now where a lot of these big models are integrating nutrients more explicitly. A lot of them are adding in phosphorus cycles for the first time, for example. And certainly I will say I am not a modeler, but I'm interested in working with modelers. And so I'm going to give a quick plug for a group I am on the steering committee of. It is a National Science Foundation, so the US, funded research coordination network, where our goal is to better integrate experimentalists and modelers, bring them together in the same room to talk about designing better experiments and better models to represent nutrient constraints on the carbon cycle. And so to give you a sort of more specific example, like model frameworks are often trying to get at what is the carbon cost of nutrient acquisition? So they maybe want to have an optimization function where plants allocate their spent carbon to, say, enzymes or to mycorrhizae or to building more roots. And in fact, there's one, this is a recently updated kind of offline version of a common model structure that has built in a nitrogen cost for phosphatase. And when they run that model, they're predicting that fixes should have a growth advantage in tropical forests because of their ability to trade nitrogen for phosphorus. And up until now, we actually didn't really have good experimental data to back that up. So the models are like, a couple steps ahead maybe of the data. And thankfully, actually, I think that our conclusions from that experiment maybe would support that decision. But we're interested in thinking about how we can address more of these kind of model structures with actual data over time. I will also add, as Alex mentioned, I started at the beginning of the pandemic. I've also been on maternity leave. I'm new to Canada. And I am really looking to build more relationships with people in your excellent department. And so there's a couple other things that I work on that I didn't mention today that I want to kind of throw out there. If this is interesting to anybody, I would love to chat with you. One is that we have got some technology that's improving our ability to measure nitrogen fixation. So not just in plants, but also in like ants or moss or leaf litter or soil, anything you can think of really. A second one is that we are also working with another big collaborative group on releasing a new global database of nitrogen fixation patterns. So not just in plants, but in things like mosses and soils. And finally, if you're interested in that kind of model data integration, the Insight Network, its membership is open to anybody. And we are currently planning some measurements of kind of plant soil stoichiometric feedbacks. We're designing a big distributed experiment that might generate some data that we can use to constrain some models. So if any of those things are interesting, please do reach out to me and we can chat about them. But otherwise, thank you for the invitation. I will leave you with some gorgeous photos from Costa Rica. These are mostly from the Osa Peninsula. I think a couple of them are from Guanacaste. And take any questions that you might have. There's plenty of time for that. So thank you so much.